or some of this will look familiar, so it'll be less for you to study overall. Um, and definitely a hot topic. This is not going away. Bioremediation probably started going big guns around in the 80s, I would say, 1980s. And it has been gaining strength because, of course, we keep polluting the environment. And the pollution's not going away. And boy, yeah, the next slide would be brighter. All right. So bioremediation was the first thing to come along. But since this class is plants, we're going to be focused in on a lot of phytoremediation ideas. And of course, anything that has phyto in the word means it's plant or plant related. And it's the idea, of course, of using plants to clean up the pollution areas, whether it be soil or water and air quality as well. So don't forget about that. Having plants in your environment, in your apartment, your bedroom, all very good. Real plants, not the fake ones, for cleaning up your air quality. The term bioremediation, that was really the first concept that came along, and it was really zooming in on the microbes. And in this class, we're talking about phytoremediation, but we can't ignore the microbes. They create a nice teamwork effort. So we will be definitely thinking about both plants and microbes. But initially, when they thought about how are we going to clean up all these polluted sites, they turned to microbes. That was the first idea. And it has expanded since then to include plants for several reasons. And then they came up with this term. They love to throw terms at us. So rhizoremediation is another term. That rhizo is root or root-like. So this is the idea that it's a teamwork effort. It's the plants. Of course, whenever plants grow, they exude all kinds of things from their roots. We say their roots are leaky and they can exude all kinds of things. And that often those things are good for the microbes that hang out there. It's good nutrition and it attracts the microbes to the roots. And it can repel, of course, certain other microbes, pathogens, and so forth. But we're going to think about this idea of the plant and the associated microbes that are doing the cleanup of these different polluted sites. What kinds of pollutants are we talking about? All kinds. Take your pick. It's a big long list. Organic. Here are some organic pollutants. I have the written out what each of these mean. Trichloroethylene, polycyclic aromatic hydrocarbons. It's in the notes section. And so on. TNT, explosives, believe it or not, can be degraded. So microplastics is the new hot topic as well as PFAS, those two on the end. And they can be degraded. It's going to take some effort and a lot of work, but all of these can be degraded to some extent by microbes in particular, but plants can be helpful. Inorganic pollutants, these are elements like heavy metals, for example. A lot of these are produced through agricultural processes, and if you overdo it, over fertilize, and things go beyond your agricultural field, you start to get polluted groundwater, rivers, lakes, oceans. Mining practices can release a lot of inorganics in terms of metals. The military, of course, with all their operations, you got that going on. Inorganics are in their simplest forms, and so you can't really like break those down. The goal with organics is that you can break them down, hopefully all the way to CO2 and water would be ideal because that is not harmful when you get to that level. But at least if you can break organics down to at least a less harmful molecule. But inorganics, they're like, lead and cadmium and zinc, things like that, where too much of it can be bad. Plants, certainly are exposed to this, of course. If it's toxic levels, they can survive, some plants better than others, and they can take up the inorganics and they can sequester them. They can tuck them away into their body parts that aren't as damaged by the toxicity, the over concentration. They can also stabilize those elements in the environment through primarily through their root actions. And so that's another option. So there's some options to still remediate or at least stabilize them. Why have so many people started thinking about using plants? One big plus is that they call it the green clean method. When you see a field of beautiful trees growing there, it's easy to ignore that those trees are being used to remediate that site. And so it looks pretty as it is doing its job. It looks pretty. So that's very attractive. And it's using plants, of course, it's environmentally friendly and it's cheap. It's usually much cheaper than other methods that can be used. For example, like taking a bulldozer in there and digging up all the soil and bringing it off site to some area where it can get processed. That's a whole nother matter. So we call what the plants are doing, we say it's in situ at the actual site. You're not taking the polluted soil away, you're just working with it there. And in general, when you do that, it is cheaper. And plants in general, buying plants to put them in there is generally cheaper. So we're going to think about how can we do this? What are the pros and cons when you start using plants? Sometimes it's a good thing because it can actually reduce other organisms' exposure. I mean, that's great if that can happen, but we have to think about this. There's some nuances to that idea that we have to think about. Phytoremediation in general is fairly easy. Plant some plants, and it's not terribly difficult to do that. Lots of options on how plants can deal with pollutants. I'm going to work through some of these ideas. This should look familiar to those of you in plant phys. It's a nice graphic showing some options. So let's look at stabilization. So you see that in the root zone where you have they're trying to show that there is a shift from a circle to a square. So they're trying to show that there is some alteration to the pollutant in order to further stabilize it so that it doesn't run off, for example, into the groundwater. You could stabilize it in a way that makes it less bioavailable. I mean, that's really the trick. If you can make compounds less, pollutants less bioavailable, that means it won't impact living organisms as much. All right, so that's one option that keeps it in the soil for thinking about soil pollutants. Phytostimulation, again, we're looking in the soil and what the roots are doing. This time we're looking at, they're showing the circle being broken down. So what they're trying to show is that unfortunately they didn't really put any microbes in the soil, but you have to imagine that there are a bunch of microbes in the soil. And so this is also called rhizodegradation, rhizoremediation, because now we're thinking about the plant root excreting substances that will feed the microbes. The microbes help break down the pollutant. So you see this very often with especially hydrophobic compounds because microbes can get at that quite nicely. Also, sometimes what the roots exude can make hydrophobic compounds more available for breakdown. So it depends on the particular pollutant. So those two methods, phytostabilization, phytostimulation, because they're below ground in the soil, herbivores that come along to either eat the leaves or maybe pollinate the flowers, they're not getting direct exposure. So that's good. So we need to think about that as we work through some of the above ground stuff. So the above ground, For phytoextraction, you see that the plant is taking up the pollutant from the soil and putting it in the leaves, extracting it from the soil. So when they take up heavy metals or other inorganics, they can do that. But of course, now it's in the leaf tissues. So if the herbivores come along and start eating that, they can get exposed to the pollutant. If it's a nasty aphid, don't really care, but if it's something else. All right, phytodegradation. Now you see the idea of a pollutant's getting taken up. Degradation, they're actually breaking it down. So we're looking at organic pollutants that the plant can actually work with and take and start running its physiological pathway and start breaking it down. And then phytovolatilization. It's pretty self-explanatory. The pollutant is taken up. It's volatile and can often go through the plant unchanged, although sometimes it is changed, degraded en route. So you don't have to memorize exactly which organics fit into which category here, but if you want an example, TCE and MTBE are classified as VOCs. And then it's not on this graphic, but another one is hydraulic control. That phrase is showing up a lot in the more recent literature. They're really big on this idea because they're realizing how powerful root systems are, and the idea that if you design your remediation efforts to have different types of root systems, different depths of root systems, that will work better. And hydraulic control is the idea that the roots are busy taking up water and dissolved molecules, right? And bringing them up. And so that means that less will end up going into the groundwater in terms of the pollutants and the rivers and lakes and oceans. So hydraulic control, using the root systems, because as you think about when plants are transpiring, they're taking in the water from the soil, the dissolved compounds, whatever they may be, bringing them up into the plant through transpiration. Processes, so that if you can get a lot of plants with different root systems, different depths, that's a really good strategy. Speaking of water, they're really looking at wetland remediation as well. Rhizal filtration, whether it be natural systems. I've seen quite a few papers published over in India where they have some pretty polluted rivers where they just plant more plants into the river and let them grow and do their thing. And they can find that that can be suitable if they pick the right types of plants. We'll talk a little bit more about that. And then the constructed wetland is a little bit more thoughtful in the sense of they're really trying to, they know a lot about what the pollutants are in the river, for example, and they design the type of plants. You can see in figure B that there's different species of plants, different locations. Some are floating, some are all the way down and with deep root systems. So there's different types there. And so they're getting fancier. The earlier papers used to be, let's just plant some plants in the river and call it good. Now they're getting fancier and getting into more of a constructed wetland idea. When you look at what types of plants, of course, you think about aquatic plants. So like cattails, for example, you tend to see those in very along rivers and lakes and ditches. You don't have to memorize any of these, but these are some examples of aquatic plants. We have a lot of Elodea or Alodia around here. Duckweed, of course, Izola, water hyacinth is a big one right now. And then poplar and willow can actually handle at least pretty wet areas, so those are also good. Poplar, the populus genus aspen, they have been used in phytoremediation for a long time. That's a very popular tree. And willow is gaining in that as well. Kind of a nice thing with azolla and duckweed. So that would be like plants that hang out. They're floating plants. So from that perspective, they're easy to harvest. So if, for example, they take up the pollutant and sequester it, you can just skim off the azolla and duckweed and harvest it that way. It's pretty easy. Often what they do, if the plant has taken up the pollutant, they'll harvest the plants and then take them somewhere else to be processed. And typically it's incineration if they know that the pollutant is still in the plant and still could be a risk for herbivores. So wetland, they use often a mixture of rooting and floating, emergent and submerged. So this is a good strategy, not putting all your eggs in one basket. Here is a paper that was published not too long ago where they created a system with wastewater. And they were acknowledging that they definitely, you think about the plants, but then you also try to promote certain microbes that are good at remediating. So this is a nice abstract showing the idea that the bacteria are going to be helping just not only with the remediation effort, but as we know, we've been learning through this class that many of the microbes are very beneficial for the plant in general and just help the plant. They're kind of like preventative medicine. They kind of help the plant thrive and survive. Here, they're looking at the oil contaminated wastewater, um, different kinds of oil. They were factoring in endophytes. You can't forget about them. We're going to talk about endophytes in remediation in a bit. But they're showing how, the roots are taking up the oils. And they're trying to show the different kinds of bacteria there. And they're showing several different ideas here, the biodegradation, the volatilization. And so they're saying by the end of the treatment, you have much lower levels. You can look at the oil levels before and then the oil levels after. We do have good results with that. So they are looking at these types of things with a lot of the lakes and rivers. Oil, there have been several oil tankers that have, broken open and spilled all kinds of oil. So we'll think a little bit about that, but it is often when you think about the microbes in particular, there's some really interesting study going on with those types of oil spills. Plants. So if you're going to pick a plant and you want it to remediate, you have to know a little bit of the plant physiology. You have to know about your pollutant. And it'd be a good idea to pick a plant that's fast growing. Typically, that means they have fast transpiration rates, which helps take up any pollutants from the soil in particular, when you think about them growing in soil. You typically want plants that produce high overall biomass, that they grow really luxuriantly, lots of good growth, because that allows them to take up more of the pollutant and stuff it into their plant body if that's what they're going to be doing. Obviously, you want them to be able to grow in the contaminated site. And so they have to be able to withstand the climate and the soil type. Their overall plant morphology, looking especially at the root structure and the depth. That's very important. One of the reasons poplar trees have been used so many years now is that they know that they have pretty good deep roots, and that has been an attractive quality. But don't forget the surface area too. The surface area is good also for allowing for a lot of microbial growth, not just for taking up the nutrients or the pollutants, but it really does support a lot of the beneficial microbes that can help with remediation. So you do want fast transpiration rates. Poplar is also known for that. So the poplar tree species, they typically have good fast transpiration rates. So in general, you're looking for that. Um, that can mean that you'll end up with volatilization potentially of the pollutant. So you have to think about your pollutant and is that a risk? Is it okay as a risk? Sometimes it is, but you do have to think about that. They've really been getting into what are the roots doing more carefully now. They've progressed quite a bit in the research, looking at what are these plants exuding to for a couple of reasons. Are any of the exudates able to help break down the pollutant or sequester it, stabilize it? And then, of course, are these exudates helping to feed the microbes that are doing the remediation in the soil or in the water? You certainly want to know what's going on with the pollutant. Where does it end up? If it's going to be sequestered above ground, you have to think about that. And for quite a while, they weren't thinking about that, but I see more and more in the more recent literature that they're really being more thoughtful about the idea of, okay, if the plant takes it up and the pollutant is still a risk, it wasn't broken down to a safer state, what is that going to do to the ecosystem in terms of wildlife that comes along that might feed on that plant or pollination going on? Do we have to worry about that? So they're looking at where does the pollutant end up? Is it stem, leaves, flowers, fruits? What's going on? Probably the main reason that they really started looking at plants to begin with way back is that way back they found out that there are plants that they termed hyper accumulators that they kind of it was a kind of a serendipitous discovery where they were looking at remediation and they found some plants in a habitat that they found out was highly toxic, lots of minerals, toxic levels of minerals and, well, heavy metals essentially. And these plants were growing in that soil, and they were surprised to see how well they were growing. They weren't super huge plants, but they were plants capable of growing through the growing season. They looked good. They started studying them and they realized that they were hyperaccumulators. They were taking up the heavy metals and accumulating them to about 100-fold higher than what other species were capable of. And when they did further studies, they found that these hyperaccumulators, it was a variety of different inorganics that could be accumulated to very high levels, and yet they survived. And so they have been studied for many, many years trying to figure out how do these plants do so well. And of course, they would like to learn, like, if we can study this system, can we then apply that to other plants and maybe engineer other plants to be as good as these hyperaccumulators? The one downside is they don't grow super fast because they're stressed. They're in an environment that's stressful and they don't produce a ton of biomass. So there would need to be some tweaking, genetic modification of some sort in order to make this a little bit more optimal, I guess. But it's a good study tool. And like I said, it was kind of a serendipitous discovery that has led to some really good research. Okay. I think this is a good spot to stop because the next topic is getting more into, I'm kind of switching gears a little bit and looking at wildlife impacts. So I'll stop with this. Why don't we take a little short break? Um, like, or yeah, you don't have to get up and move around if you don't want to, but let just a few minutes so that we can get set up for the next thing. Okay. Whoops. Okay. All right. So with phytoremediation, there's some extra concerns that you need to think about as opposed to if you're just using microbes. And that is what's going to happen with the wildlife that interact with those plants as they're remediating. If they take it up into their above ground, especially their above ground body, then definitely you can have wildlife and pollinators impacted by that. In general, a general rule of thumb is that grasses tend not to accumulate inorganic pollutants in their shoots to any, huge amount. But at least in the studies they've been doing, dicots can do that. So grasses, by the way, those are monocots. Dicots tend to accumulate more. And here's a good quote. If a toxic volatile pollutant is emitted by plants, the fate of the gas in the atmosphere should be determined as part of risk assessment. Great idea, but that was often overlooked with earlier research that was going on and remediation efforts. They didn't think too much about what's going on if it's volatilized out of the plant primarily through the stomata. So this is a new thing, relatively new. So volatile selenium and mercury, some of the studies published, they said, yeah, it doesn't pose a threat. That's what they're saying. But I don't know. We'll see. I think more studies need to be done, but at least the published paper that was done, this was back before 2005. So it was a while ago. They're claiming that selenium and mercury volatilization was not seriously at a concentration that was serious enough. Um, you know that with regard to universities, we're not allowed to have mercury anymore. Like, the old thermometers are mercury-based thermometers. Like we had to get rid of all our thermometers. Did you ever break a thermometer and watch the mercury roll around? When I was a grad student, I broke more than one mercury thermometer and yeah. So back then it's like, whatever, just watch it roll around a little bit and then sweep it up. And but yeah, we had to get rid of all of that. That's why all the thermometers here now have the red. I don't even know what that is. Does anyone know what that red substance is inside the thermometer? It's some solution, but I don't know what it is. It's not mercury anymore. It's not the silver mercury. Did you ever it ethanol? Have you ever heard the expression mad as a hatter? You're mad as a hatter. That means you're nuts, insane. And the reason that expression came about, from what I learned, is that when people way back in the Victorian era made those black top hats for men out of beaver pelt, you would dip it in a solution of mercury as part of the processing of the pelt. And it was usually like a family business, like father and son would run the business. And they noted this trend of fathers going insane and then the sons would go insane in that family. And they finally linked it together. It's because I don't know what you call people who make hats for a living, but they were in that business and they finally traced it to mercury. And that mercury was causing the insanity. And so mad as a hatter is mercury exposure. So, I mean, they were exposed to huge amounts of mercury over their business lifetime. So I guess it's not too surprising. So the idea of pollinators, and we all know bumblebees are really, having some issues with colony collapse disorder and that whole mess going on. They're threatened from that angle. And now with all the pollution that has been going on, a lot of plant biologists and entomologists are getting very worried about what is this doing to our bumblebee population. They're already at risk, and now we may be hitting them with heavy metal accumulation. So they're starting to look at this more seriously and do some nice studies with it. And This was interesting. They were looking at aluminum and nickel, and they found that aluminum didn't seem to mess with the bumblebees. But the nickel, if the flowers had nickel in the nectar of the flower, the bumblebees seem to be have an aversion for that. They weren't visited, the flowers weren't visited as much, and they just didn't like the nickel in the nectar. And so they, from a distance, of course, the bees can't pick up on any heavy metals in the flower. It's not like it gives off, well, as far as we know, it's not like it gives off anything that they could detect either through ultraviolet light or detection through any of their other senses. But once, of course, they do take up the nectar and feed, then they might be able to actually detect what's going on. So they're looking at kind of parsing out these different metals and testing them against what's going on with the bumblebees. And of course, they're also looking at what types of heavy metals end up in flower tissue and in fruit tissue. So they're looking at that more carefully. And so they're saying, okay, if we need to think about this, if we're going to use angiosperms in particular for phytoremediation efforts, we do need to do enough research to think about the flowering, or well, either don't grow the plants to the flowering stage, or if you are going to grow them to the flowering stage, you definitely need to think about what's going to happen to the nectar. Are the metals going in there or the other pollutants? And so this is not something that's been looked at historically and is something I would say from about the 2010 onward is when people started to get more serious about studying it. And, again, the idea of hyper accumulating plants, if some of that really high level of the heavy metals in particular are ending up in the flower tissue, that could be a real problem. Certainly, you can see bioaccumulation of heavy metals through the food web. That has been documented. So that is something that even, so the bumblebee gets it, maybe they're not super affected by it, but then whatever eats the bumblebee might be affected and whatever eats that and so on and so on. And so the food web idea, certainly you need to consider it. The ramifications kind of ripple out from just the plant taking up the heavy metal or the pollutant. So they've done some nice studies with predatory insects and arachnids, looking at them attacking the bumblebees, for example, and other pollinators. So they're saying, especially when it comes to the bumblebees that are already at risk, this is something that really needs to be carefully thought about. Some really nice studies are looking at, especially when you get the entomologists and the microbiologists involved, they've been looking at bumblebee physiology. And this paper I thought was really a good paper. Cadmium and selenate exposure affects the honeybee microbiome. So the microbes that hang out with the honeybee and metabolome. So when they say metabolome, it's all the metabolites that are being made by the microbes as they hang out with the bee. And the bee-associated bacteria show potential for bioaccumulation. And so when they did this was a 2019 paper. So they're saying the honeybee microbiome is mostly resilient when it comes to the cadmium and selenate. But it's kind of a case by case basis. So they were talking about the core symbionts. So when you look at the microbes that hang out with any organism, there's going to be different percentages of those different species within the organism. So they have to look very carefully at, who's all there in terms of the microbes and which ones are the core symbionts versus the lower population levels of, the kind of the minor players of the microbiome. And so they were able to show that some of the bacteria that come out with the honeybee could accumulate the cadmium and a little bit of selenate. And they actually said this is good as long as the microbe is hanging on to it and not exposing the bee to it. So in a way, it's kind of like the bee is sequestering it through its microbiome, which I thought was a nice angle to think about. And so they're saying it could provide a protective mechanism against the pollutant if the microbe can take it and either sequester it or, well, with cadmium and selenite, you can't really break it down because they're already broken down to their simplest forms, but detoxify in the sense that the bee wouldn't get direct exposure. So this idea of I really like that they're getting at the bee's microbiome and wondering if that can really help. And, if they could find some really nice microbiome strains that hang out with the bee, hopefully they would think about exposing, getting those into the bees population so that the bees can get a leg up on. They're already hit from so many different angles with regard to the colony collapse disorder, and just they're not getting very good nutrition when all they get to feed on is a monoculture crop. That makes it very difficult for them to get a diverse meal, so to speak. They're only getting, it's like eating a Big Mac every day. They're just getting one thing when they go and pollinate monoculture egg fields. So that was a nice study. So you definitely have to think about, where's the pollutant going when the plant's interacting with it? Is it staying below ground? Is it coming up into the above ground? Is it getting broken down? Do they have any enzymes to break it down? Do they have transporters in their cell membrane to bring in the pollutant? Are they volatilizing it? And so one thing to think about, they say that all those organic pollutants that I was listing out on a previous slide, they're saying those are xenobiotic to plants, which just means that plants didn't evolve in the presence of those, at least in any high amounts of those materials. And so they never evolved ways to take them up, really. I mean, it was not something that they evolved to do because they weren't exposed to it until humans came along and did all the pollution. And so generally, they're not finding any cell membrane proteins in the plant cells. So when you look at all the transport proteins that allow substances in and out of the plant cell, you don't really find transport proteins for these organic pollutants. And that means you have to rely on diffusion for those pollutants to get into the cell, and then whatever happens after that point can vary. So that is something to think about because that will slow down the rate of what's going to happen to that pollutant once it gets taken up into the plant body. So this is a pretty nice graphic showing kind of the difference between what a plant is doing with organic versus inorganic and some options that they have. So here the artist is trying to show that with the organic pollutant, they don't show any transport protein here. What they're trying to show is that it's just coming in through diffusion. And then once it does come in, they can adapt. They do have enzymes that can break down some of these organic pollutants. And so they're trying to show that they can physiologically act on the organic pollutants. Another thing that they can do is called conjugation, which I talk about this in Plant Phys a fair bit. With conjugation, that just means they add something onto the molecule. And so they're trying to show that with the GSH, and you don't need to know what the molecules are. It's often a sugar of some sort. But what they do is they add on another molecule to the pollutant, and that kind of inactivates it and detoxifies it. It doesn't make it reactive to the plant. And then they're showing that an option is to sequester the pollutant. If it's acted on or not, they can still sequester it in the cell wall. So the cell wall is a good spot to put pollutants because it's relatively inert. There's no sensitive chloroplasts in the cell wall or mitochondria that can get impacted by that pollutant. So the cell wall is a good strategy. And then here with the inorganic pollutants, you're seeing they do tend to have transporters that can bring in the pollutant. And again, you can sequester it, conjugate it, the vacuole. So I would like you to know the vacuole and cell wall. Those are big relatively good strategies because the vacuole is large. It's the largest organelle in the plant cell. And it's not the nucleus, it's the vacuole. And so you can stuff a lot of stuff into that space. And then the cell wall is usually pretty built up nicely too, so you can put more stuff in there. So physiologically, they have options once they bring in the take up the pollutant, and just kind of varies a little bit between organic versus inorganic. Soil systems, I'm not going to go into huge discussion on this, but I think most of you who have had a soils class, you know soil systems can be extremely complex and highly variable, depending on where you're looking. You have anywhere from silt and clay to sand in terms of size range of particles of the soil. You have all the chemical properties to think about, including the pH, of course. We know plants love pH 5.5 to 6.5. Pollutants, some pollutants do really well and being highly soluble in that range, others not so much. Remember that soil particles are negatively charged, so that is going to play a role. If you have some of the heavy metals that are positively charged, for example, like, their ability to stick to the soil particles and be there as a reservoir. Don't forget about cation exchange too, where you can have these positively charged pollutants potentially getting knocked off the soil particles into the water solution. So we've talked a little bit about cation exchange earlier in the semester, and you've seen this diagram already. So just think about, especially with the inorganic pollutants, if they're positively charged, they can be on the soil particle, and through cation exchange, they can get released off the particle into the soil water and then taken up. And so if you are trying to remediate on purpose, you want this plant to take up a certain heavy metal that's positively charged, then you need to know a bit about cation exchange capacity and work the system as best you can to get cation exchange happening so that you can get the positively charged pollutants up, off the soil, in the water, and up into the plant body. So there's a lot. This is a very interdisciplinary production. When you think about phytoremediation, you definitely need plant biologists and physiologists involved. You need microbiologists. You need soil chemists involved. And so one of my previous grad students, MC, she got her master's here with me, and then she wanted to travel for a while, and that was the next step she wanted to do. So she got a job in Chicago with a phytoremediation, bioremediation company, and they flew her all over the United States. She loved it because she got to fly all over the place and go to these contaminated sites. And it was her job to assess the site and take samples and bring it back to the business in Chicago. And then that's where they had a whole team of, the plant physiologists, microbiologists, and the soil chemists, where they would, run the materials, the samples, and try to figure out what to do. So she liked the job because it was fun to travel and go and interact with these different people. And of course, she loved the idea of remediating these environments, but it was very stimulating because she got to work with a lot of different scientists with different levels of expertise. So it was a good job that she had for a while. And then after that, she went on for her PhD for a while, after several years of that. So you've had this picture before. Just remember, the plant really needs to have the pollutants in the water of the soil for them to easily take it up. That's why microbes often, are good at helping remediate, because they can actually release the metals off of the soil particles themselves. They're really good at that. So we talked about previously this idea of the Lyotrophic series, how if you read it from left to right, the way you interpret these series is that the thing on the furthest left has the highest binding affinity. And so this means in this particular case, aluminum, if there's enough aluminum in the soil, it can bump off all the other cations to the right of it. And then, so for example, and then like the hydrogen, the proton, H plus, it can bump off all the ones to the right. And so you just keep interpreting that way. The binding affinity is high to lower. And so understanding the soil chemistry definitely comes into play when you're working with heavy metals and understanding how to maximize the situation where you can get the plant taking up these minerals, these, well, pollutants. So typically with the inorganics, they're present in charged form. And so they can get into the water, they're hydrophilic, and be brought up by the plant quite readily if the cation exchange and the microbes are doing their job. If the pollutants are hydrophobic, that's a different thing to think about, because if they're in the soil, they're not going to want to interact with the water very well in the soil. So they tend to be bound to the soil particles and not want to come off of it to mix in with the soil. So they've had this issue when they're trying to remediate, for example, PCB and PAHs, which have been around for a long time. And they've realized what they need to do is either apply surfactants themselves, the humans will go in and do that, or I put bio in parentheses because you can also get microbes to do this for you. If you pick the right microbes, there's microbes that are really good at doing this. And if you can get that situation cooking in the soil, you can have that as well. And so there's been both cases with remediation efforts where they'll go in and put like a soapy substance, a surfactant, kind of like a soap, is what we're thinking here. And so they've done that where a human will go in and do that, but then they realize too that microbes just naturally do this. That's how and remember, when microbes are, like if microbes are releasing granulolipids, for example, and they're trying to release PAH off of the soil particle, what are the microbes doing with it? They're breaking it down and using it for nutrition and energy. So believe it or not, they are breaking down pollutants. It's good for them because they're getting their needs met. They can get their ATP and often their carbon sources from that. So it's something that can harm us is actually feeding them. So it's pretty amazing. And then just remember, if you have volatiles, you have to really think about that because sometimes they're acted on by the plant and sometimes they just go right through the plant out into the atmosphere. Okay, we're going to stop there. We're over time. Thank you for your patience. And so Wednesday, we will wrap this up. And then I'm hoping to use a good chunk of Wednesday, like the second half, especially to talk about the final exam. Okay. Oh, and one more presentation too.